This is the main PrESSTo page at PrESSTo.BINF-KU-DK. The page contains information and links related to the human promoters and enhancers. What we want to do is look at use cases for enhancer and promoter search and discovery. From the main PrESSTo page, we can navigate to the human enhancers and promoters selectors. We will start with an enhancer example. This is the main enhancer search page. There are a total of approximately 33,000 robust enhancers. See main enhancer publication on robust enhancers. This number of enhancers will change as we define our search constraints. As presented in the background research video, we define constraints like at least 30% of the total expression from this enhancer should come from, for example, brain. We use sliders to define such constraints. We can observe that we have two panels of sliders. The left panel contains the primary cell samples and the right panel contains the organ or tissue samples. Both panels influence the resulting enhancers, but their sliders are independent. What this means is that, for example, in the organ panel, if we set the constraint to at least 30% for brain, we cannot have more than 70% for eye. Notice how when we change the brain and eye slider values, the resulting number of enhancers immediately changes too. In the case of eye, we cannot increase more than 71% because we have reached already 100%. On the other hand, we are free to set other constraints for primary cells even if we have reached 100% for tissues. In both panels, we can set the sliders to at most 100%. One can observe the remaining percentage indicators. Of course, in this case we have been too restrictive in our search and there are no enhancers that qualify. Let's reset our cells and organ sliders and show a use case scenario. Suppose we are interested in blood and we want to have both white blood cells and red blood cells. We will use neutrophils for white blood cells and reticulocytes for red blood cells. Let's set the slider values to around 40% for both neutrophils and for reticulocytes. We only obtain one enhancer and we would like to have more than one. Let's decrease the slider values to around, say, 20% for both primary cells. In addition, we want to have most of the tissue expression also from blood, say around 80%. This search results in 3 enhancers. Also, we could have searched by genomic location, but we will show this in the promoter case. We can see more information about our 3 enhancers by clicking on the See Detailed Results link. We are now on the Results Viewer page. On the left side, we have the list of found enhancers, in our case 3. Given that we have searched for blood and blood cells related enhancers, we will describe the results page while at the same time analyzing the first of the 3 enhancers. For each individual enhancer in the list, we display related information and available actions in the tabbed panel. In the first tab, we have expression-related information. As specified in our search, we see that most of the organ expression comes from blood. In the case of cells, we have around 40% from reticulocyte and 40% from neutrophils, along with other immune-related cells. In the second tab, we have the option of viewing our enhancer as a track in the UCSC genome browser. We will keep the default values for the enhancer track and go to the genome browser. In the genome browser, we can see some of the enhancer properties specified before. H3K4 methylation 1 and H3K27 acetylation marks, but not H3K4 methylation 3 marks. DNA sensitive, a number of chip transcription factor peaks. It is also slightly conserved. Since we are looking at FANTOM 5 enhancers, they have already been defined to have bidirectional cage peaks. See main enhancer paper. In addition, the enhancer is located on the intron of the IL-12RB1 gene that binds interleukin-12, which is part of the immune response. This is good to have since our search included blood tissue and neutrophils and the enhancer was expressed in a number of different white blood cells. If we move to tab 4, we can see if the enhancer expression is correlated with promoter expression over the cell and tissue samples. We can observe that the enhancer correlates with the number of anonymous FANTOM 5 promoters along with FANTOM 5 promoters associated with known genes. It is good to see that an IL-12RB1 associated promoter also correlates with the enhancer. Among others, we can also see the JAK3 gene, which is predominantly expressed in immune cells. In the last section of tab 4, we can check if there are any SNPs overlapping enhancers. In this case, we have a number of SNPs for which we can view more details by clicking the links in the last column. In tab 3, we can view the enhancer in the FANTOM 5 Zenbu genome browser, but we won't go into details about this. In addition to individual enhancers, we have the option of performing actions for all the resulting enhancers. We can download the enhancers as a bed file, view the enhancers in the UCSC genome browser, get the DNA for the enhancers, and perform sequence motif discovery using MEME. This concludes the enhancer use case, and next we will move on to a short example on promoters. From the main PrESSTo page, we can navigate to the Human Promoter selector. As we can observe, the promoter search page is almost identical with the enhancer search page. The only difference is that we can input the strand of the promoters we are interested in, whereas for enhancers we don't have strandness. Also, there are many more promoters than there are enhancers. In the promoter case, we will search for promoter locations close to known genes. Suppose we are interested in the gene NeuroD1. As we are writing the gene name, we get suggestions for the candidate genes. After selecting our favorite gene, we see on the left where the gene is located. By default, we extend the up and downstream locations by 100 kilobases. For our example, let's have 0 bases downstream and 1000 bases upstream the gene. In total, we get 11 promoters close to NeuroD1. We will also like to sort the promoters by their expression in the neural stem cells. We click on the See Detailed Results link to view information about the promoters. Similar to enhancers, we have the promoter list on the left and the tabbed panel containing individual promoter information and available actions. In the case of promoters, we can also check if they are anonymous or associated with known genes. The promoter P1 at NeuroD1 is associated with the NeuroD1 gene, as we expect. The promoter expression is related to brain and eye for both tissue and cell samples. Just as with enhancers, we can view the promoter in the UCSC and Zenbu genome browsers and check if there are any overlapping enhancers and SNPs. In addition, promoters can be analyzed with the FANTOM 5 Explorer tool, but we won't go into details in this video. As for the operation on all resulting promoters, we can download as bed files, view in the UCSC genome browser, get promoter sequences, and perform motif discovery using MEME. This concludes our promoter use case example. In conclusion, we have shown a few things we can do with PrESSTo. For further information, please visit our About, Contact, and Documentation sections. Similarly, relevant links are included in the description of this video. Thank you for your interest in PrESSTo.